Hello, everyone, and welcome to today's webinar titled Developing Saliva-based Lateral Flow Immunoassays. This webinar is a part of the 13th Annual Clinical Diagnostics and Research Virtual Event. I am Antonina Salcido of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Fortis Life Sciences. For more information about Fortis Life Sciences, please go to fortisislife.com. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. You will also notice that the speaker has provided a survey located to the right of the Ask a Question box. This survey will be available during both the live session and the on-demand period. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. I now present today's speaker, Andre Alfaro, Senior Director of Assay Development at Fortis Life Sciences. For a complete biography on our speaker, please visit the Presenter tab from the menu at the left of your screen. Mr. Alfaro, you may now begin your presentation.
Hi, everyone. This is Andre Alfaro, the Senior Director of Assay Development for nanoComposix and Fortis Life Sciences. And today our objective is to go through saliva-based lateral flow immunoassays. And what we'll be talking about is the three major considerations that everyone should be thinking about on the onset of those projects, and really the details around being successful through that process. So before we get into the saliva-based lateral flow immunoassay development, I want to go and discuss a little bit about who we are as a company. So Fortis Life Sciences was started about 2.5 years ago now, really going and identifying founder-led companies and acquiring those to make a family of companies as a one-stop shop for life sciences. And really associated with product developments in this space. So you have companies like my company, nanoComposix, who specializes in nanomaterials, therapeutics, and assay development and manufacturing, as well as Empirical Biosciences, which is specific to enzymes and proteins and custom manufacturing, as well as some excellent antibody companies in Bethel, Arista, and Abcor. Really, the idea here is when any component, whether it's a reagent or services associated with the product development in this space, we want to be your help or help you with anything you need. And with that, I'm going to jump into saliva. Why saliva? Really, this question has come up quite often recently as we push more to this over-the-counter market, which COVID and COVID products have really shined a light on the utility and the effectiveness of rapid testing in the OTC POC space. So why saliva though over a nasal swab, urine, other matrices that are in this OTC space already? Really, it's because of the reasons on the left of this slide, which is ease of access. So lots of sample readily available, non-invasive, meaning that there is no Lancet or collection method that people typically have an issue with. Acceptance by patients, really that's the evolution of saliva testing in general, starting with OraSure, which we'll talk about momentarily. Reduced risk of infections because there is no needle or anything associated with that collection. And it still has the accessibility to common biomarkers that are really targets for a lot of the rapid testing space. A lot of those are infectious diseases. These can also be nucleic acids, growth factors, inflammation markers, really anything you can think of. If it is in serum or in blood, you're typically going to find it in the saliva. Now, with that in mind, what comes next? So you have a target, you have a matrix. What is the steps and what are the considerations when making a product? And that's really what this slide is about. So what are those considerations? And for me, at least, and for our services here at nanoComposix, we tackle this question in 3 different phases. There's your sample collection, which is the physical collection of that saliva. How are you collecting it? With what? How are you controlling for volume, sample normalization, and those kind of things? Then there's the sample processing. How are you going to make a sample or a filtered sample that's going to achieve what you need to for sample or sensitivity to that sample, analytical sensitivity. It could be dynamic range, it could be time to result or assay kinetics. There's a lot of considerations that processing is going to be directly tied to. And the end of the day, it also has to do about result interpretation. Like, how are you going to interpret those results? Is that going to be quantitative, semi-quantitative, qualitative? You're going to use a reader or no reader? What is that metric you're going to use for result interpretation? So this discussion today, we're going to break it up into those 3 topics and tackle each of these considerations separately. So I wanted to provide a level of context for the discussion, really an example of a successful saliva diagnostic in the OTC space that you could use as a backstop to some of the things we talk about, like sample collection and transfer and processing. So I want to take a quick look at the Orshore test. This is one of the first at-home saliva test. This is an HIV test. And you can see really quickly, looking at the components of the test, why it was really effective in this space and how it got approval. When you go OTC, that means reduced complexity in order for it to be really approved by the FDA. And that's because there's a lot of human error involved in sample testing. And if you don't want a trained operator doing this in a CLIA lab, then you do need to simplify the toll test method. And that goes back into your considerations of, what kind of product you want to make and where is that test going to be performed at. Really changes how you look at sample collection and normalization and such. But I'm going to make an assumption that if we're talking saliva, we're talking in the OTC space. And the Orshore is a great place to start when you're looking at, what is it that I'm trying to make. So quickly going at this and how, those 3 considerations I just mentioned. So starting with sample collection. So they use a single test device that has a sample collection built in to the strip. So swab at the end, or sample pad or sample collection pad, goes into the mouth, collects that sample, which wicks up the stem and into the strip. So your sample collection and sample processing is occurring there. And included with that sample processing is the solution vial. So this helps extract that protein so it doesn't bind and stick to the sample pad before it hits the membrane. So they have some extraction or running buffer that helps with the flow, helps with the collection process. And that's something that will also have to be considered as part of your product development. And ultimately, how is that test read out? It's a visual test, and usually using 40 nanometer gold right at that test line. But taking a step back, it's relatively simple, right? It is your saliva collection method that connects to the nitrocellulose membrane and the conjugate pad, which is in a pouch with a desiccant. So you need a Mylar pouch, a desiccant, either combined or separated saliva collection device and the test cartridge, usually an extraction buffer. And then in this case, to reduce the added steps to the end user, there is not a sample dropper tube that you have to move sample from place to place. It is collect and then drop in the vial. These are all the considerations really you need to think about before you start. So once you get started and focusing on the sample collection, there is 3 different considerations with regards to the sample collection. Evaluate the collection methods. There's multiple types of collection methods. We're going to talk about that. Whether you're doing passive drool, stimulated saliva, active collection. There's different methods to collect, and we're going to talk about those. Understand that what is available commercial, that OTC space for collection devices. We're going to talk about some of those companies in a moment. And then, and the third option, after you've taken those two into account, is a customized pathway. And there are companies and CROs that will develop custom methods of saliva collection, whether that is, a nylon swab or a cotton swab, and what and how it's attached to what length of a stem. Is it a breakable stem? All of those things can be customized. So, sample collection. There's two common sample collection methods. There's passive drool and an oral swab. Now, these are two very different things with different pros and cons to each. So, passive drool, very common. This is as simple as, and then the image kind of indicates that, where an individual is functionally spitting into a collection device, non-filtered, very simple, very inexpensive, because this is just a funnel and a tube. So these polypropylene tubes are very inexpensive in bulk. The funnels are inexpensive. You're looking at maybe 25 cents or less combined. There is no paper-based material, so there's no loss of material, which is a very common issue with any of the current swabs. Even the OraSure, right? So they use an extraction buffer to help reduce retention of analyte in the swab. High volume capacity, right? You can build big tubes if you want to do testing on PCR as well as the test for external testing. There is a high volume capacity capable with this method. And it's relatively simple. It's easy for someone to understand. Please spit into a cup. But the cons, there are a number. So some samples, particularly in the ICU, you'll have patients that can't drool or can't spit. Whether that's because of the ailment, or in any case, or age, it can be difficult to do this. Lots of variabilities, and this goes hand in hand with viscosity. You don't control, that matrix very well when it's just spit. If someone's dehydrated and there's not a lot of water content and it's more phlegm, then it's harder sample matrix. So there's little in the terms of control and consistency with this method. Now, on the other hand, you have the oral swabs. And these are going to be more consistent because there's a process that goes into the cons. You do have to follow a procedure to do this, kind of like what this image is indicating right now, that you're swabbing different locations. There are over 2,000 salivary glands in your mouth. Where you're swabbing, under the tongue, on the cheek. Are you using an abrasive? Do you want to stimulate saliva? All of this can be done with the swab, so it's really important on the procedural end, but it's going to be very consistent. This is because there's a level of filtration involved, whereas there's none in passive drool. There's quite a bit, depending on the material, the bed volume of that swab, and the duration on that level of sample normalization and filtration you can get. It's resistant to viscosity variability, so because it's a filter, it's waiting for a specific bed volume of that liquid phase. Some of these saliva collections have indicators on them once a certain bed volume has been hit, so you can have more of a fixed volume for collection. And there's a lot of options for this. So the OTC landscape is very broad. Now the cons, like I mentioned, sample or analyte retention in swabs has to be examined. Your analyte versus my analyte, you're going to have different reactivity to different materials. There is no one size fits all when it comes to the right material. This is a common mistake people make, that they fix to, brand X because it's what they use on a different product. You really have to look at your spike in recovery using these swabs and determine whether that retention is an issue or not. And you always have solutions like what OraSure did with their extraction buffer. It doesn't have that kind of volume capacity of kind of the passive drool collection. So you only get what that swab can allow. Now you can have larger swab collection volumes, but that can get maybe 1 to 2 mils, but it's not going to be nearly as much as passive drool. And like I mentioned, the procedural importance. This isn't something that you want people to sporadically swab their mouth. There's usually a specific location you're targeting, whether that's under the tongue for X amount of time, roof of the mouth, cheeks, these are or throat. These are things that you have to take into consideration. So these are the different collection methods. I don't want to limit it to these two, but these are the two major ones that you should think about. And that goes right into this conversation on what that landscape looks like for products you can purchase. Now, this is just three very common and some of the bigger names in this space. That's Celimetrics, that's Oasis, and that's Porex. All of which do kind of swab development, saliva collection. They have a wide variety of ELISAs that might help with examining method comparisons and recovery. So they have a lot of tools that can help with saliva. Now, before you go down this path, because some of these devices can be quite expensive, you want to think about what your specific test system is going to look like, what that cost point you need to achieve is, and really start understanding what that sample retention is going to look like. So three considerations again with this space. Do you need how is a swab going to work in your system? OraSure determined an independent swab is not going to be the right path. That's too many steps. So we're going to I need to integrate that. They went down a custom route, which is different than what we're talking about here, but it could be an integration of potentially both. Can we add, one of the Oasis saliva collection devices with a test strip in a cassette? Sure, that's just customization. So lots of options here, but I wanted to get you guys started on kind of where to start looking for those swabs. Now, we talked about the different methods of sample collection. So let's talk about sample processing. It's really hard to have that, conversation independently than the sample collection, because the question you need to ask is, do you want the sample processing to occur at the same time as your sample collection? That's that passive drool versus active swabbing. So sample processing considerations. Filtration and separation, right? So this is kind of what we talked about, using a swab to do it. Now, for quantitative tests, for example, in saliva, passive drool is going to be very difficult. It's going to be very difficult because of that variability. Depending on your test platform and your system, you want that filtration to occur as early as possible, or have a level of filtration occurring all the way at the sample collection portion. That can be done really through the swab itself, or if you do have passive drool, you can push it through a filter on the device itself, or a different metric, but you do need to somehow extract and separate that liquid phase from the mucin. And the mucin is really the major problem. It's going to cause some really significant issues when it comes to viscosity and sample flow. That's why we recommend, for quantitative tests, we don't have the ability to handle some variations in sample consistency, like a qualitative test may. For quantitative, that sample has to be consistent every time. So we do recommend using a swab allows some filtration ahead of time. And that might not be the only filtration you have. It could be filtration with the swab that goes into another, say, mesh filter that's built into the cassette to extract that liquid phase. So that's something to consider. And if you look at some of the over-the-counter options from the Oasises of the world, you'll see that they have almost like syringes and plungers that you can collect a saliva. You have a saliva swab that goes into a mouth that you put into what looks like a syringe, and it goes through another filter before it's dispensed. That's the level of filtration possible before it even touches the cassette. Because you really want to resolve this as soon as before it hits the cassette, in a lot of cases on the standard design, because it just won't absorb into the sample pad. And the viscosity will be to a point where the absorption in the liquid phase is going to be too slow. That means your assay kinetics are all going to change. So you got to be very careful of that. Another solution to sample processing is dilution. One of the older solutions to this, and now there's a lot more on the filtration separation side. You don't always have to just jump to dilution, and the reason why I say that is because dilution it adds complexity. Not that it's complicated, but for an end user, depending on your space, it can be. A lot of the markets for that where the saliva is so valuable is in that OTC space. Asking, patients or users in the field to take 2 drops from your saliva or take that swab, mix it into a extraction buffer, pull out the swab, and drip that in, very similar to a COVID test, is possible. And that would be the consideration initially. But again, that's not fixed volume, right? So you have to be very careful that pathway is only suitable for qualitative tests. And if you want to do quantitative, then dilution becomes even more tricky because it's only a fixed volume into a fixed volume in order to achieve that, calibration you've built into the product. And the other solution for sample processing, so we talked about filtration, whether that is through a filter post swab, or that's dilution of the passive drool or the swab itself. The third option is additives. These are salts, these are surfactants, these are proteins. These typically are dried in the sample pad, but these are only as good as the viscosity of the solution that's going into it. So salts are great for normalizing things like pH. We all know that antibodies work and function at preferred pH ranges. So getting each sample to be at the certain pH is going to be critical. So you almost always have to have some level of salt somewhere in your assay to bring it to, say, pH 8. Surfactants are going to be critical to flow, so they help block non-specific binding through your sample pad, conjugate pad, onto the membrane. So really important to sample flow and specificity. Proteins for blocking, for example. Saliva is a snapshot of, your blood, but it comes with a lot of constituents and proteins and enzymes. So it's important that we have some level of blocking involved somewhere. And again, typically in the sample pad, but there are creative ways to do this as well. You can lyophilize these in tubes that the sample gets put into before it gets dropped into the device, and so forth. And really, ultimately, it's a combination of all of these things. It's not necessarily one or the other. It is a combination of all. So we've walked through the first two, and now we're on kind of result interpretation. So there's two methods for result interpretation. There's reader-based and visual. Reader-based are great and required if you're having fluorescent tags, where this is, whatever fluorophore you're using. If you're using europium, for example, you're going to need a reader. There's lots of good off-the-shelf options for this. One I recommend for a kind of a low-cost multi-use reader is from ChemBio. They have something called the Cube, very customizable. Price point's around $300. Great for this space, unless you have an on-strip reader or a semi-disposable reader, that means somewhere between 25 and 50 scans, perhaps. The ChemBio is unlimited scans potentially. It's capable of doing quantitative. So these readers are capable of doing quantitative and semi-quantitative readouts. Integration with the cloud and healthcare system. So that's another benefit. It can also screen the results if you don't want the end user to be interpreting those results and they want to go to a clinician, then you kind of need a reader to do that. It is sensitive to price though, mind you, because you have to, that $300 is going to have to be either a one-time fee you're asking customers or broken up and embedded into the cost per test. And there is some uncertainty around the regulatory route here. You have to ensure that for the OTC space that your reader is part of that submission, right? And for a lot of companies that wanted cell phone-based readers, this is kind of the hurdle they're jumping through right now is that regulatory route with the cell phone-based reader. Visual is very simple, and it is really binary, and you can do semi-quantitative if you're not really in that OTC space and is going to go to a CLIA lab. There is some wiggle room on semi-quantitative, but realistically, this is a qualitative method. It's either yes, no. Great for something like COVID, for example. We're going to get a visual test line, and the absence and presence of a line tells you. And really, taking COVID as an example, you don't really care about the intensity of said line. You just want to see if there is a line and that is present. And the intensity has to do with that semi-quantitative nature. Do you have a lot or a little, and what that means? Typically, the FDA doesn't like us or like an untrained operator making a determination of what a lot and a little is and what that changes into their, behavior. That's why there's no IFU that says if your line is really intense for COVID, you're at higher risk than if it's a lower one. It's like, no, if you have a line, you have a line, and that means you're positive. That is for certain products. And for infectious disease, it's a very worthwhile to not overcomplicate it, but there's multiple paths. So visual, there are quite a bit of materials out there available to help with that readout. Now, for anyone who's been in this industry for a while, you know that 40 nanometer gold and gold colloid is the godfather of nanomaterials associated with lateral flow. Lateral flow really became relevant in the 80s, and since then, 40 nanometer gold has been a staple of products. You can see this very distinctly in most, if not all, pregnancy tests. But there's been a lot of evolution in this space, mainly because the need for better and more sensitive particles is becoming more and more common, particularly when you're getting to matrices like saliva. Saliva is great because of all the things we talked about earlier. The ease of collection and non-invasive and wide array of proteins available. But the issue will always be that they're never going to be in the highest concentration as they are going to be, say, in blood. So how are you going to get there? How are you going to get that same sensitivity, but with a non-invasive matrix? Well, one of the solutions is, in fact, the nanomaterial. And really, that's what nanoComposix really got into this space, into the saliva and lateral flow space, was in the nanomaterial, was the nanomaterials, and specifically with the gold nanoshells. So gold nanoshells aren't required for all assays. 40 nanometer gold, it's less expensive, it's robust, and really can achieve the job. But if you are trying to achieve certain sensitivities and you do want a visual readout, because you want to go in that OTC space, or just because you want a higher sensitivity test, I do recommend, looking at gold nanoshells as an option in this case. And this just has to do with the fact that these particles, by virtue of physics, are just brighter and more intense. That's because they're physically larger. So these are 150 nanometers versus 40 nanometers. And as you can imagine, you see bigger things better because they can absorb more. But typically, and anyone who's worked in and made gold before knows that larger particles really have issues on flow characteristics in lateral flow. 100 nanometer gold colloid or 150 nanometer gold colloid won't flow very well, if at all, on lateral flow. Why is that? Well, it's because the density, and then they're functionally like little boulders rolling around. And what we did is hollow that out to a degree. So we have a silica core, so very light, not very dense, and we shelled that with gold to give it the plasmonics of large gold, but the density of something much smaller. And that's what we call gold nanoshells. So these come in both passive and covalent options. So sometimes the sample variability can be an issue with passive absorption. So I want to make sure there was an option with the carboxyl modified surfaces, so you can do covalent conjugation. But really, I just want everyone thinking about what is it that they need? What's the cost point they're willing to hit? What's that quality, and what's the characterization associated with that? So 3 very important characteristics associated with this. Now, I wanted to look at some examples of gold nanoshells, in saliva, looking at common markers, and this is an example of nanoshells with 40 nanometer gold for troponin I. So troponin I is a very common marker for cardiac distress. We want it to really indicate and show a difference between what nanoshells can do and versus 40 nanometer gold. Typically, depending on what your desired concentration range is, you might be perfectly fine with 40 nanometer gold. You can see the sensitivity down there from 0.5, to 30. Well, what if you needed it less than that? What if you wanted something along the lines of, 0.05 nanograms a mil? Well, Fortis don't really have that, but you can get that with nanoshells. And so we do recommend taking a look, depending on your sensitivity and product needs, the nanoshells in this regard. And just because I love using our SEM here in nanoComposix, I asked for an image of what the nanoshells look like on the strips themselves. And that's what you're looking at here. So this is an SEM image of nitrocellulose membrane, and that's the actual strip that was used. So you can see that intensity of a line, this kind of blue-gray. What does that mean in terms of line? Is it completely packed? Is there any more binding? How big are these nanoshells versus the pore structure of the membrane? So this is in an MDI 150 membrane, and you could see the nanoshells, or the kind of white dots on the nitrocellulose themselves, and how much additional packing space you have. So as intense as this line is, we're still not fully packed in terms of space on that membrane. So that's kind of the value proposition. And any publications around nanomaterials in the cancer space and infectious disease or parasitic disease, we wanted to add in some references to periodicals about specifically these. So I do recommend taking a look if you want to get going on procedures and materials. These publications are a great starting spot. And another example too is in the serological space, which became very prominent for COVID, looking at immunizations and vaccine efficacy with these serological tests. And so nanoComposix developed a saliva serological test looking at IgG content in saliva using oral swabs. We actually made this semi-quantitative. Now, I mentioned, reader systems in this space. Well, this is never designed to be OTC, but we wanted to show that you can go semi-quantitative. So we have a scorecard that the intensity of the line is compared against. But really, you can mirror side by side serum and saliva. I mean, that is what this graph here is showing, is that these are kind of right on top of each other in terms of correlation with one another. So now, again, though, you can see how much higher it is in serum versus saliva. So unless you have the right particles and develop the right systems looking at retention and collection, you got to be careful. So that's another consideration. So you can mirror, blood, and specifically serum in this case, to saliva, but there's got to be a lot of considerations in mind when you're doing that development. And then at the very end, assay design and manufacturability. So we talked about sample collection, we talked about sample normalization, and we talked about result interpretation. And those are considerations for development. But don't forget to consider manufacturing at the very end. Kind of the never develop a product that is not manufactured or at a cost point that is not sustainable for reimbursements or cost or price to the customer. So early as you can, we recommend determining your target price. You should have that in mind when you're even looking at, Oasis and Porex on swabs. Don't end up buying a $12 swab or saliva collection device when your product cost point has to be under $2. Always keep automation in mind. What can you automate? As early as possible, identify those kind of components, whether that is, buffer filling using liquid handlers, whether that's web handling systems for conjugate spraying and nitro spraying. Always keep that in mind so we can find methods to keep your cost points down. And like always, consider your supply chain. Don't get bogged down with suppliers that you can't control or have long lead times. This is a consideration that many forget until the last moment, and it is something that they should be considered. And really looking at that in terms of that manufacturability, I love this example of a rapid diagnostic in the COVID space, because really the COVID space being OTC, cost point was everything. How can I get this under $2? How can I get it under $1? Now this is an IFU for an EUA approved COVID test, very similar to those that many have seen. And everything here is basically designed to be automated or low cost. And this is how you get those cost points. So, there are tubes where, heat sealed with, it looks like a Mylar top. Okay, so those plastic consumables are, pennies. thus heat sealing and the dispensing of that buffer with precision of plus or minus a microliter, and the heat sealing of that. These machines are very inexpensive. These are about ranging from 25,000 to 200,000, but at the low end, these things do about 2,500 a minute. It can dispense, seal. Their next step, the swab, independent swab, these that come in sterile packaging. Those are very low cost depending on where you're going and what kind of retention your swab has. So again, low cost. Tubes that were designed and transferred. So it was their extraction buffer was in their dropper tube. So it's not another piece. So again, they consider this across the board for what they were trying to accomplish. So kudos to them and the development team for making something that is going to be very low cost, because the test strips themselves can be fully automated. They assemble the cassette, fully automated, and then they made a buffer tube that's automated, and then just the consumable in terms of the swab that's purchased sterile and added to the kit. So I want to end on kind of the manufacturing capacities here at nanoComposix, and this is really associated with exactly what we're talking about: integration of automation into your process. So that's what we do here, whether it's the web handling systems for dispensing on, sample pad blocking treatments, conjugate on your conjugate pad, antibodies under nitro. That is all done on kind of automated systems. So we can do roughly here about 7 million units a month. And here's a good example, one of those liquid handlers. So bowl feeder orients the tubes that goes into a system that ejects fixed volumes, goes through a heat sealer and stamp to get that foil on it, to use about 2,500 an hour. And then lamination can either be whether it's reel, so this is high throughput, or card-based. So either way, there's options for different volumes depending on your needs. And really just ending back on kind of where we started with the companies and what we do. So just to summarize everything before we talk about the companies, 3 considerations when it comes to saliva: sample collection, sample processing and normalization, and result interpretation. And it all feeds into manufacturability and cost points. So nothing here is done in a vacuum. All of those have lots of gray area and overlap. So all must be thought about together as well as independently. And then coming back of how we and our company can help, this is kind of a summary slide of that. So on the protein side, we have Bethel, Arista, and Abcor. Whether these are enzymes, proteins, is quite large in terms of the array. Nanoparticles, that's really going to be nanoComposix. So that's the gold nanoshells we talked about. That's the 40 nanometer gold. If you're looking at different conjugation systems using, streptavidin gold and biotin, protein A particles, we sell those as well. And really any of the enzyme, again, the enzymes and recombinant proteins, that'll be empirical. So, and finally, just a summary of kind of the registration, where we're at with this. So we have, empirical in San Diego are ISO 345, are both FDA registered. We have sites across the country with various capabilities. So with that, I will leave it, and don't forget to let me know if you have any questions about this presentation, about saliva, about lateral flow, about assay development in general. I'm happy to help. So thank you, everyone, and I look forward to hearing from you.
Thank you, Mr. Alfaro, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsors, Fortis Life Sciences, for sponsoring today's webinar. Questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until November 9th, 2024. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with any one of your colleagues who may have missed today's live event. Until next time, goodbye.